Hello everyone, welcome to our channel. In this video, we are going to discuss about the Mendelian disorders, one of the very important topic in pathology and genetics. So, what are Mendelian disorders? They are mutation in single gene, with large effects. What are mutation? Mutation are the permanent change in the DNA. So, Mendelian disorder can be recessive and dominant. There are certain definitions that we need to understand before going deep into the Mendelian disorder. First is codominance. When both the alleles of a gene pair contribute to the phenotype. Phenotype is a clinical expression of any genetic disorder when the both alleles of a gene contribute to the phenotype. For example, in histocompatibility and blood group antigens, they follow a codominance. Another definition is pleiotropism. When a single mutant gene result into the many clinical implications, which is seen in sickle cell anemia. Genetic heterogeneity. When the mutation at the several genetic loci, mutation in several genetic loci will represent as a same trait, just opposite to the pleiotropism. So, genetic heterogeneity seen in profound childhood deafness and diabetes mellitus type 2. Transmission pattern of single gene disorders or Mendelian disorders. Mutation in autosomal gene, which can be recessive or dominant, and mutation in X-linked gene, which can also be recessive, and some of the X-linked disorders are X-linked dominant disorders. So first we are going to see is autosomal dominant disorders, manifested in a heterozygous state. The capital A represent a normal allele and a small a represent an affected allele. So this is the pedigree chart in which a heterozygous state in a parents. Another parent is absolutely normal, and this will result into the 50% of the affected offspring and 50% are normal. Characteristic of the autosomal dominant disorders. With every autosomal dominant disorder, some proportion of patient do not have affected parents. Some of the parents could be normal. Mutation either this is seen in when the mutation is in either egg or sperm. Clinical feature can be modified by variation in penetrance and expressibility. So autosomal dominant disorder follow incomplete penetrance, which indicate some individual carry mutant gene but are phenotypically normal. They have mutant gene but there is absolutely normal phenotype, which is seen in incomplete penetrance, a feature of autosomal dominant disorders. Variable expressivity. Mutant gene in the trait form expressed differently in various individuals. Many conditions in which age of onset is delayed, like seen in Huntington's disease. The molecular mechanism of autosomal dominant disorders depend on the nature of mutation and the type of the protein affected. So this is a brief about autosomal dominant disorder. Now we will discuss about the examples of autosomal dominant disorders or common diseases where we see this kind of genetic defect. Autosomal dominant disorder. Here we discuss system by system. In the nervous system, neurofibromatosis, Huntington disease, myotonic dystrophy, and tuberous sclerosis. In the urinary system, polycystic kidney disease. In GI, familial polyposis coli. In hematopoietic system, hereditary spherocytosis, von Willebrand disease. In skeletal system, Marfan syndrome, Ehlers-Danlos syndrome, some of the variants of Ehlers-Danlos syndrome, osteogenesis imperfecta, achondroplasia. In metabolic disorders, hypercholesterolemia, acute intermittent porphyria. So these are the examples of autosomal dominant disorders. They are very important and so many questions asked from these disorders. After autosomal dominant disorders, we are going to discuss about the autosomal recessive disorder. The largest category of Mendelian disorders are autosomal recessive disorder when both the alleles of a gene locus are mutated. They express in homozygous condition, characterized by the various features. Trait usually does not affect the parents, but the sibling may show the disease. Look, if A is a normal gene and small a is affected one, then this condition is absolutely normal. Both the alleles are normal. If one is affected, then this is known as trait. If both the genes are affected, then this is known as a homozygous condition. This is trait and this is an homozygous condition. So, in autosomal recessive disorder, trait usually does not affect the parents, but siblings may show the disease. Siblings have 1/4 chance of having the trait and the increased likelihood in the consanguineous marriages. Features distinguish autosomal recessive disorder from the autosomal dominant disorder. Expression of the defect tend to be more uniform than the autosomal dominant disorder. Expression is usually uniform. As we have discussed, in autosomal dominant disorder there is variable expressivity. They follow autosomal recessive disorder follow complete penetrance, unlike the autosomal dominant disorder where there is incomplete penetration seen. Onset is frequently early in life. They are rarely detected clinically in heterozygote condition. Mutant gene encodes for the enzyme in the heterozygote condition. 50% of the enzyme is normal and 50% of the enzyme is defected, and usually there is no clinical manifestation in heterozygote condition or the trait condition. Now we will discuss about the various examples of autosomal recessive disorders. We will discuss them by system. In metabolic disorder, cystic fibrosis, phenylketonuria, galactosemia, homocystinemia, lysosomal storage diseases, alpha-1 antitrypsin deficiency, Wilson disease, hemochromatosis, glycogen storage diseases. In hematopoietic system, sickle cell anemia and thalassemia. In endocrine, congenital adrenal hyperplasia. In skeletal system, Ehlers-Danlos syndrome type 6 and 7C. and alkaptonuria. In nervous system we have neurogenic muscular atrophy, Friedreich ataxia and spinal muscular atrophy. So these are the examples of autosomal recessive disorder, another very important topic for the NEET PG MCQs. Now we will discuss about the X-linked disorders. In the male, as we know, male has the genotype of XY, male only having one X chromosome, so they express as a male is hemizygous in X-linked disorder. Basically, an affected male does not transmit the disorder to his sons, but all the daughters are the carriers, as seen in this pedigree. This is an affected male. All the daughters of the affected male is all the daughters of affected males are having the disease and males are normal. So these are the various pedigrees and the various combination. This carrier female, in carrier female there is a chances of carrier daughters and affected male. In a diseased female, both the daughters are carrier and both the male are affected. Likewise, we have the various combination of this pedigree in X-linked disorders. Now we will discuss about the various disorders in X-linked recessive and X-linked dominant disorders. X-linked recessive disorders. In musculoskeletal system, Duchenne muscular dystrophy. In hematopoietic system, hemophilia A and B, chronic granulomatous disease and G6PD deficiency disorder. In immune system, agammaglobulinemia, Biscott-Eldridge syndrome. In metabolic disorder, diabetes insipidus and Lesch-Nyhan syndrome. Look, there are so many types of mnemonics you can find, and I'm suggesting that you make your own mnemonic for better memorizing power. In X-linked dominant disorders, we have vitamin D resistant rickets, Alport syndrome, Reed syndrome, Fragile X syndrome, incontinentia pigmentii. So this is it in Mendelian disorder. Thank you.